All right, welcome you guys. We are going to finish up molecular biology today. So last week we talked about DNA and DNA packaging along with DNA replication. So today we're going to talk about DNA transcription and translation and a little bit about gene regulation. So transcription and translation is really what we're going to focus on today. So how do we read our DNA, turn it into a message in the form of RNA, and then how do we translate that into amino acids and proteins? So essentially, how do we read our DNA and create proteins out of it? So that's the whole process of DNA transcription and translation. And then we'll talk a little bit about gene regulation. How do we turn genes off or on? And how do we regulate which ones are producing proteins? So last week we talked about the first function of DNA, which is replication. And we undergo DNA replication during the cell cycle, during interphase. So we have to replicate our DNA to be able to undergo cellular division, and whether that's mitosis or meiosis, we need to replicate the DNA first. And that's making an exact copy of our DNA so that we can split it into 2 or 4 different cells, just depending on if we're doing mitosis or meiosis. So the second function of DNA is to actually provide the blueprint or information to make proteins. And this is where we're going to find transcription and translation. So we read our DNA, right? Or another word for it is transcribing the DNA into a messenger RNA molecule. So that's what M stands for, messenger. So messenger RNA or mRNA. And then we use that mRNA molecule to provide a code for the proteins. So essentially it's like a secret code, and then we read that code and build our proteins. And a protein is just a polymer of amino acids. So essentially there's going to be a code for each amino acid, and we're going to chain them together and create or build a protein. And that part is the translation, right? So we're going to translate that messenger RNA into the building blocks for a protein. So both eukaryotic and prokaryotics are going to undergo the fundamental process of transcription. So eukaryotes, right, we have a nucleus, and so we're going to undergo transcription within the nucleus, and then we're going to take that messenger RNA out of the nucleus and actually translate it in the cytoplasm, okay? Whereas in the prokaryotes, we don't have a nucleus, right? So no nucleus. So we actually undergo transcription and translation in the cytoplasm, right? So that's the difference between eukaryotes and prokaryotes, but we're essentially going to undergo a similar transcription and translation process. Mostly transcription. Translation is a little bit different, but we'll talk about that. So what's the first step in DNA transcription? Well, we're going to have to initiate transcription. So just like we talked about in DNA replication, we're going to have to unwind the DNA. Now we had to unwind pretty much all the DNA to undergo replication, but now we're just going to unwind part of it. Okay, so we only need to unwind a part of the DNA to undergo this mRNA synthesis. So this is done within a transcription bubble. And there's actually an enzyme, just like we had an enzyme in DNA replication. This enzyme is called RNA polymerase. Okay, so we're going to be creating an mRNA molecule from our DNA. And how do we know where to start? How do we know where to unwind the DNA? Well, we actually have a promoter region on the DNA template strand that says, hey, start here. This is where you're going to start transcription. And that's where we are initiated for DNA transcription. So then the RNA polymerase molecule knows that this is where I'm going to go to create the mRNA. So once we have initiated transcription, we're going to elongate the messenger RNA, or elongation as our second step. So essentially the RNA polymerase is going to produce a complementary strand of RNA to our DNA template strand. So it's very similar to DNA replication, but instead of producing a DNA strand like we do in replication, we're going to be creating an RNA strand. So that's why we have our RNA polymerase. And so we have to remember in RNA, we are replacing thiamine with uracil. So wherever we would have had a T, we're going to have a U. Okay, so we just have to keep that in mind when we're making our RNA messenger molecule. So now we have our last step in DNA transcription, and that's termination. So we have to stop transcription. So the RNA polymerase actually stops once it hits this termination signal. And a termination signal usually is a repeating nucleotide sequence. So essentially it just repeats itself so that it says, okay, I know to stop, and I'm going to release from the DNA. Now in prokaryotic cells, and that's the picture I have below, once transcription terminates or ends, translation is going to be happening at the same time. So we have lots of ribosomes that are going to be then translating that mRNA right away. So the picture shows you just a strand of DNA, which is the green, and then the mRNA is going to be in blue. So we've created a little mRNA strand off of that DNA template, and then the little red dots are ribosomes. So we just say it's a polyribosome because there's multiple ribosomes all sticking on that mRNA and translating it at the same time. So we'll talk more about translation in a minute and the ribosomes, but just know that in prokaryotic cells, transcription and translation are really happening kind of simultaneously. So that's going to be a little bit different in our eukaryotic cells, right? So we'll talk about the differences there. So in eukaryotic cells, we have to actually transport that mRNA out of the nucleus. After it's been transcribed, we need it to be translated in the cytoplasm. So that's different from those prokaryotic cells where everything is happening in the cytoplasm already, so it's kind of happening simultaneously. So something we need to do before we exit the nucleus is we actually have to splice out some information that is added that we don't actually really need. Okay, so it's kind of interesting. And you don't need to worry too much about exons and introns, but essentially it's going to do a couple of things, okay, before it sends it out. And what happens is it actually adds some ends on either side of the mRNA. So on the 5' end, it creates a little cap. And then on the 3' end, it creates this poly A tail. Okay, so just as a repeating sequence of A's, and essentially that's going to add some protection for that mRNA molecule. So and again, we're not going to get too much into introns and exons, but essentially the exons are what are coding for proteins, but the introns don't actually code for any sort of functional protein. So essentially we kind of cut them out, and that's RNA processing. So we have to do two things. We have to process the RNA by cutting out those introns, and then we have to put a 5' cap and a poly A tail onto it before we can tell it to, or before we can allow it out of the nucleus. So this is RNA processing. So once we've processed that RNA, once we've protected the ends and spliced out all those introns that we don't need, we are going to go outside the nucleus, and then we're going to be translated. So DNA translation, also known as protein synthesis, right, is really the most energy consuming process in the cell. This is where all of our energy is really going. And proteins really make up the majority of all living organisms. If you kind of exclude water, because obviously we're mostly water, but other than water, we're mostly protein. And so what we're going to do is we're going to translate that mRNA, or decode that messenger RNA, into a polypeptide product, which is going to just be a string of amino acids. So essentially that mRNA is going to code for all these different amino acids, and we're going to string them together and build a protein. So I have a great video on transcription and translation that you can check out either through the PowerPoint if you can click on the link, or I also have it on Canvas, so definitely check it out. So what are the structures that are required for protein synthesis or DNA translation? So obviously we're going to need that messenger RNA template, right, that we've just built from our DNA transcription. We're going to need ribosomes, right? So the ribosomes are what are going to build the proteins. And within that ribosome, we have what's called tRNAs, and these guys are transfer RNAs, and essentially they're the ones that are going to bring in the different amino acids, and we'll talk about that, and you can better understand it, I think, in the video as well. We also have other enzymes that are going to help us that we're not going to cover too much, but there are other enzymes that are involved in protein synthesis. So now we're going to actually read that mRNA. We're going to translate that mRNA. And how do we do that? Well, we use what's called the genetic code. And this is a universal code. So essentially, for virtually all species, the mRNA is going to translate into the same amino acids. So that's why it's a universal code. And it kind of stems the theory that, you know, all living things are really coming from the same place because of this universal genetic code. So each amino acid is defined by a 3 nucleotide sequence. Okay, so 3 nucleotides in a row are going to be called a codon. So one codon is going to tell you which amino acid is going to be next, essentially, or going to be strung up in the sequence. Okay. So how do we do that? So this little table on the right hand side, and you can see a lot of these tables, you can look up the genetic code. And so this is a simplified version of it, but again, it's a universal code. So essentially you take the first letter of the sequence on the left hand side. So there's really only 4 options, right? We have 4 different nucleotides. So you either have a U, a C, an A, or G. And then you take the second letter, and that's the middle section, and then on the right hand side you have the third letter. And then if you notice in the middle, you can see that multiple codons might read for the same amino acid. So say, from on the left-hand side, second one down for leucine, you see that there's actually 4, 5, 6 different sequences or codons that actually will encode for leucine, that amino acid. So there are some that have lots of different sequences and some that are a little bit, maybe only 2 of them code for that amino acid. And we do have one start codon, so that's not in this table, but you can look it up. So we do have one codon that actually says start, right? So it doesn't actually encode for for an amino acid, but it says this is our start. And then you have multiple stop codons. Okay, so they're in red on this table to show you that this sequence means to stop. Okay, so we have some start, we have one start and a couple of stops. And then all of these different sequences code for all of these different 20, right? We have 20 amino acids, so every amino acid has a specific codon or multiple codons. So what is the actual mechanism of protein synthesis? And again, I think the video is going to really help understanding this process, okay? So what happens is we have these tRNA molecules that are going to come into the ribosome. So they're going to come into the ribosome and they're going to contain an anticodon sequence, which is essentially the corresponding sequence to our codon. Right? The codons live on the mRNAs and they are going to code for a certain amino acid. And so the tRNA is actually going to have that amino acid attached to it. So the anticodon is going to recognize its codon on the mRNA and bring in that correct amino acid. So then that correct amino acid is going to build on a chain of multiple amino acids, right? So it's just going to go, the ribosome is going to make its way down the mRNA, bringing in the tRNAs that are going to match the codons, right? And then they're going to create a polypeptide chain of amino acids, right? And what happens is that these mRNA molecules are so long that we actually have multiple ribosomes translating at the same time. So that's similar that we talked about in our prokaryotic cells, but essentially in those, the transcription and translation are happening at the same time. Right now, we've already done the transcription in the nucleus, and now we're outside the nucleus, and the translation is happening by multiple ribosomes all at the same time. Okay, so not just one ribosome is chugging away through the whole mRNA. We actually have multiple ribosomes working on it. Okay. All right, so I know we went through some complex processes fairly quickly, but I want you guys to take the time to watch the videos and go through these processes to really understand what's going on. But I want you to really take away the big pictures as well. So I don't want you to get too caught up on all the details, but I just want you to kind of get the big picture. So let's do a little bit of review on that big picture. So go ahead and pause the video and take your time answering these 2 questions. So, well, 4. So what are the 3 steps of DNA transcription? And what's the end product of DNA transcription? And what makes that product? And then what is the end product of DNA translation and what makes that? Okay, so go ahead and pause it and we'll go through the answers. Okay, so hopefully you were able to take a crack at these questions, and hopefully it'll kind of solidify the big picture. So our 3 steps, right? So we have initiation, right? We're going to initiate it with that promoter region. We're going to unzip that DNA and start creating an mRNA molecule. So that is our end product, right? We have an mRNA or messenger RNA molecule, and what makes it is the RNA polymerase enzyme, okay? So this is similar, so don't get it confused with DNA replication, right? So we're creating DNA in DNA replication, and it's the DNA polymerase molecule. Okay, so here we're creating mRNA, so RNA with the RNA polymerase. Okay, so what is the end product then of DNA translation? What are we producing? We're producing a polypeptide, or AKA a protein, right? And what's making that is the ribosome. Okay, so just big picture is the ribosome, even though we know that those tRNA molecules come in with the amino acids, reading those codons, but essentially it's the ribosomes, right? So the ribosomes are going to what are going to be creating those polypeptides, those protein molecules. So now that we know the process of transcription and translation and how we actually read our DNA and produce proteins, all organisms and their cells have the control over the transcription and translation process because they may or may not need that protein, right? So that is called gene expression if we actually turn on a gene to produce a protein. So we want that specific protein, we're going to turn on that gene or express that gene, and we're going to produce that protein. But how much do we produce of that protein? And that's the regulation process. So we have to regulate how much of the protein we're actually going to produce. We can't just turn it on and leave it going, right? So then when do we stop? And that is gene regulation, right? So gene expression is turning on that gene to produce a protein, but then we have to regulate it, right? We have to tell it when to stop. So we do have cells that are very specialized in multicellular organisms. So if you think about all the cells in the body and what their jobs are, right? So if you think of different tissues, we've got very different cells in, say, the nervous tissue in our brain versus muscle tissues or muscle cells in our muscle tissue, right? So they do different jobs and they're highly specialized for the jobs that they do. And this is due to gene expression. So we turn on certain genes and turn off certain genes. So in one cell, right, maybe the muscle cells are all about contraction, right? Whereas nervous cells or neurons are all about, creating impulses and electrical signals. So very different jobs. So there's different genes that are turned on in those different cells. So this is all due to gene expression. Now, in terms of gene regulation, the control of the gene expression, when do we turn it off and how much of those proteins do we need? It's a very complex process and we have a lot of gene regulation. But there are problems, right? We do get malfunctions in gene regulation, just like we've seen malfunctions in our cellular division and those control systems. And they can be very detrimental if we have malfunctions in gene regulation. And there's a lot of differences between our prokaryotic and eukaryotic regulation. Because if you think about the complexity between a eukaryotic cell and a prokaryotic cell, eukaryotic cells are much more complex. So we're going to have a lot more regulation in a eukaryotic cell versus a prokaryotic cell. So let's compare the differences between prokaryotic and eukaryotic cells when we're talking about just transcription and translation as well as gene regulation. So if we look at the differences, prokaryotic cells really, they lack a nucleus, so they're much more simplified, right? So they lack a nucleus, and therefore we said that transcription and translation is going to occur almost simultaneously in that cytoplasm, right? So that's going to be different than our eukaryotic cells, where transcription is taking place in the nucleus, and we have to go through a processing of that RNA before it actually exits the nucleus. So if we think about it, there's a lot more steps in the eukaryotic cell, right, than the prokaryotic cell. And so this allows for a lot more regulation on the level of gene expression in the eukaryotic cell. Versus the prokaryotic cell, there's really only regulation at the transcription level. So there's really only kind of one area where they can regulate that gene expression. So lots of differences between the two. And again, we're not going to go through the details of gene regulation and the process of it, but just know that there's a lot of different levels that we can have regulation in the eukaryotic cell. And that's really due to the stepwise process of transcription happening in one location and then translation happening in another location instead of simultaneously. So this is just an image to show you a little bit about the different levels of regulation when we're talking about gene expression in eukaryotic cells. So again, this only has to do with eukaryotic cells because we really only have one level of regulation in prokaryotic cells. So if we go through transcription and translation, essentially at every step through transcription and translation, we have some control. So when we transcribe our DNA into the mRNA molecule, we have transcriptional control at that level. And then when we process that mRNA to be able to get it out of the nucleus, we have some RNA processing control, right? So that's where we add the caps and the tail, and we splice out those introns that we're not going to use. And so we have some control there. And then we also have to transport that mRNA out of the nucleus and into the cytosol. So there, during that RNA or mRNA transport, we have some control there as well. And then once we're out in the cytosol or cytoplasm, we're going to translate that mRNA into protein, right? So now we have translation control as well. So lots of different steps and levels for control, and hopefully it's going to help us find mistakes, right? And not let mistakes go through. But of course, it's not a perfect system, so we do get malfunctions, which can be very detrimental to the body and to the cells. So just to finish up with a little review, I know these aren't on your notes, but you can write them in. So go ahead and pause the video and answer these questions. And pretty much we just went through it, right? So, but these are the big picture ideas I want you to get out of gene regulation. So what is the difference between gene expression and gene regulation, right? Because we've talked about both. And then what is the difference between gene regulation in the prokaryotic cell versus the eukaryotic cell? Okay, so hopefully you were able to pause the video, write down the questions and maybe some answers. So what is the difference between gene expression and regulation? So gene expression is the turning on of the genes, right? So it's deciding what proteins we need and therefore which genes we're going to express. Now the regulation has to do with the gene expression. We're going to regulate that expression. We're going to say how much of that protein we're going to produce, and when are we going to stop producing it. Okay, so we're regulating gene expression. Okay. Now, what is the difference between that regulation in prokaryotic cell versus the eukaryotic cell? Well, we only have really one level of regulation in the prokaryotic cell, and that's really at transcription. Whereas we have many levels of regulation in the eukaryotic cell between transcription, RNA processing and transport, and translation. Right? So about 4 layers or levels versus only 1 level. Okay, so that makes sense. So go ahead and review the videos as well. I think they really help to paint a good picture about what is happening during transcription and translation. And if you have any questions, please let me know. All right, I'll see you guys later. Thanks.